After almost a year living in a mad world with no familiar faces, going nowhere, and wishing for normal birthday parties, now it's time to sit and listen. Sit and listen. To get vaccinated or not to get vaccinated? This is a question a large section of the population is considering based on doubts of the fast approval of different vaccines that attempt to decrease the transmission of COVID-19. Worried about getting the COVID-19 vaccine? Tune in as we interview scientists and doctors for more information.
According to the McGrath portal, there are 11 vaccines that have been approved in the world. There are 4 main types of vaccines being administered in different countries for the prevention of COVID-19, which include RNA, inactivated, non-replicating viral factor, and protein subunit. Let's start with RNA vaccines. Nucleic acid vaccines use genetic material in the form of RNA to provide cells with the instructions to make the antigen. In the case of COVID-19, this is usually the viral spike protein. Once this genetic material gets into human cells, it uses our cells' protein factories to make the antigen that will trigger an immune response. The proteins made with mRNA instructions activate the immune system, teaching it to see this spike protein as foreign and develop antibodies and other immunity weapons with which to fight it. There are 2 main brands that have been produced mRNA vaccines. The CDC official website and the New England Journal of Medicine released safety and efficacy reviews articles on the mRNA vaccines. Both had mild to moderate side effects in the first 7 days of vaccination, especially after the second shot, and had the efficacy values based on the number of symptomatic patients. Pfizer has 2 shots given 21 days apart in the muscle of the upper arm, which showed a 95% efficacy in clinical trials among people of diverse backgrounds and underlying medical conditions. On the other hand, Moderna had 2 shots given 28 days apart in the muscle of the upper arm. It showed a high efficacy of 94.1% in clinical trials among people of diverse backgrounds and underlying clinical conditions.
We can evaluate the risks of the vaccines using the information in both the CDC and the New England Journal of Medicine. For short-term effects, both report mild to moderate side effects as seen in these graphs.
The second type is non-replicated viral vector. In a non-replicated viral vector vaccine, another virus that cannot copy itself carries the pathogen genes. This vaccine cannot create new viral particles. Instead, it produces an antigen. This antigen is a substance that causes the immune system to respond and produces antibodies. There is a new development of suspension cell lines, which will allow the vaccine to be produced in large bioreactors, allowing the production in a large scale. 1 dose of vaccine includes billions of copies of the vector, but the vectors do not replicate, which reduces the immune response. Thus, it requires more doses to achieve longer protection. As far as safety is concerned, this vaccine is very safe and it has a low risk of inducing the disease. There are 4 brands of this type of vaccines that have been approved: CanSino At5-nCoV, Gamaleya Sputnik V, Serum Institute of India Covishield, and also AstraZeneca AZD1222. The efficacy rate of these vaccines are 91.6% for Gamaleya Sputnik V, 90% for Serum Institute of India Covishield after the second dose, 65.7% for CanSino At5-nCoV, and 63.09% for AstraZeneca AZD1222.
Inactivated virus vaccines are usually made by exposure of virulent virus to chemical or physical agents. For example, formalin or beta-propionlactone in order to destroy infectivity while retaining immunogenicity.
According to the Lancet, infectious diseases inactivated vaccines need 2 doses. The symptoms may vary from mild to moderate, and they can be given to people with compromised immune systems. The estimated efficacy is 80%. However, according to the BBC, the efficacy of Sinovac's vaccines varies significantly depending on the country where the clinical trial took place.
The last type is protein subunit. According to the Vaccine Safety Basics of the World Health Organization, protein subunit vaccines only use the protein from the pathogen. The components of the pathogen are not alive. Protein subunit vaccines only use the antigenic parts of the pathogen. The antigenic parts cause a protective immune response. They must be examined closely to find which combinations will produce an effective response with the correct pathway. A response can often be elicited, but the immunological memories might not be formed correctly, meaning that the immune system may not recognize the antigen in the future. Protein subunit vaccines are often manufactured by using living organisms such as yeast and bacteria. It must be ensured that cross-contamination with other organisms does not occur. To make it, the genetic code for the antigen must be inserted into the cell of yeast. The yeast cells are then put into fermentation tanks, and the antigen is harvested. The protein is then combined with other components to make a stable vaccine. The immune response to this vaccine is not as strong as live attenuated vaccines. However, it is safer and more stable. The Precision Vaccination website describes the process of creation and application. The vaccine relies on chemically synthesized peptide antigens of SARS-CoV-2 protein conjugated to a carrier protein and are served on an aluminum-containing adjuvant. The EpiVac Corona vaccine contributes to developing protective immunity following 2 intramuscular administrations spaced 21 to 28 days apart. Only one brand of this type of coronavirus vaccine has been approved, FBRI EpiVac Corona in Russia, according to the state trial register as reported by the TASS news agency. This vaccine is 100% effective. This is only based on a small sample size. We cannot confirm how accurate these results are due to the lack of information from the government website and primary sources.
Now let's interview an FDA representative to learn about the approval process.
Like all medicines, COVID-19 vaccines are first tested in a laboratory. This studies to check first the side effects in lab tests and animals. Then vaccines are tested in human volunteers in what's called clinical trials. These tests further help confirm how the vaccines work and, more importantly, to evaluate their safety and protective efficacy. There's no need to worry. The FDA sets rules for the 3 phases of these clinical trials to ensure the safety of the volunteers. Researchers test vaccines with adults before they do with children. Let's now dive into the development of the vaccines. To start out, companies first make small batches of vaccines, also known as lots, and they do small-scale studies to characterize and optimize the production process. They do these studies to determine suitable conditions that can keep vaccine components stable to the end of its shelf life. Also, to assure the vaccine meets its intended quality profile and complies with standards, the company develops an effective quality control strategy. Studies on pharmaceutical quality look at the individual vaccine components, the final formulas to be used, and at the whole manufacturing process in detail. The vaccine developer then conducts more studies in lab models using in vitro studies or animal models, also known as in vivo studies, to show how the vaccine triggers an immune response and works to prevent infection. Finally, the vaccine developer studies the vaccine in these 3 phases of trials with increasingly larger numbers of volunteers in each sequential phase. According to the CDC, the FDA requires that vaccines undergo 3 phases of clinical trials with real people before they could be licensed for use in the general public. So starting with the phases. Phase 1 trials are small with usually only 20 to 100 volunteers and last only a few months. The purpose of Phase 1 is really to evaluate basic safety and identify very common reactions. Phase 2 trials are larger and involve hundreds to a few thousand participants. These studies last anywhere from several months to 2 years and collect additional info on safety and efficacy. Data gained from Phase 2 trials can be used to determine the composition of the vaccine, how many doses are necessary, and a profile of common reactions. Phase 3 trials are the next step, unless the vaccine is ineffective or causes health problems, and are expanded to involve several hundred to several thousand participants. Usually these trials last several years because the vaccinated group can be compared to those who have not received the vaccine. Researchers are able to identify true reactions. If the clinical trials show the vaccine is safe and effective, the manufacturer submits a biologics license application to the FDA requesting 2 licenses, one for the vaccine and one for the production plant. During the application process, the FDA reviews the clinical trial data and proposed product labeling. In addition, the FDA inspects the plant and goes over manufacturing protocols to ensure vaccines are produced in a safe and consistent manner. Once the FDA is satisfied about the vaccine being safe, it's licensed for use to the public. Although the safety concern does not stop there, the safety continues with CDC and FDA monitoring after approval. Phase 4 safety monitoring and research begins after a vaccine is licensed and recommended for public use. The Advisory Committee on Immunization Practices, or ACIP for short, continues to check on vaccine safety and effectiveness data even after the vaccine's routine use and may change or update recommendations based on that data.
Even though there isn't information provided on vaccines that did not succeed to design and administration, the Oxford AstraZeneca has been evaluated in South Africa. The American Association for the Advancement of Science describes that the numbers are particularly low because a variant of the virus in this country is able to dodge key antibodies. This demonstrated the constant scrutiny and research the vaccines go through in Phase 4 and the evaluation of a different vaccine for the population in South Africa.
We understand that you may still be having some doubts. So let's now address some of the myths and facts concerning the vaccine. Myth number 1. The vaccine was approved too quickly. Steps must have been skipped. False. Although the vaccine was created and approved in record time, the phases in clinical trials were done according to FDA guidelines. The emergency state of the world focused research funding, technological advances, and manufacturing to speed up the process to create the vaccine and administer it to the general public. Myth number 2. If everyone around me is immune, then I don't need to get the vaccine. False. Vaccines do not just protect you, but also your entire community. There are some people who cannot get the vaccine due to their own medical history. By getting the vaccine yourself, you reduce the chance of the spread of the virus, helping those who cannot protect themselves. Myth number 3. The FDA agencies respond to the government instead of protecting Americans from unsafe products. False. The FDA has guidelines and processes necessary for the approval of a product. The COVID vaccine's approval do not respond to the government. Instead, they must go through all the phases of clinical trials and quality assurance of the product, and it is independent from any elected officials or administration.